Now we can move towards our next topic, that is basic techniques. The major developments in the field of gene manipulations were in early 1970s, when some scientists were able to genetically modify organisms, and all these developments were due to the availability of modern gene manipulation techniques. Because now scientists were able to transfer the gene to an organism like Escherichia coli, and procedures were also available for the cutting and joining DNA molecules. And these cutting and joining reactions were monitored by using a different type of procedure. But before going to discuss all these techniques, we will focus that what are the basic techniques that nowadays we are using in the subject of gene manipulation. Now, briefly, we can also focus that why before 1970s, scientists, they were not successful to transfer the gene in eukaryotic or prokaryotic microorganisms or even higher organisms. What were the basic problems or reasons? तो जो मेजर प्रॉब्लम थी वो ये थी कि देयर वाज नो मैकेनिज्म टू डिटेक्ट द फॉरेन जीन, कि जो हमने जीन इंसर्ट की है हाउ इट शुड बी डिटेक्टेड. देयर वर नो मैकेनिज्म्स. सो if the DNA it is inserted, then of course it can go inside the microorganism, but where it is integrated, no mechanism was available. And if it is not integrated, then how it may be detected? So detection was the main problem. And if the detection of the foreign insert or the DNA, it is based on the expression of the gene, then the failure of expression may be due to the inaccurate transcription or even the translation. So that's why the foreign DNA that was not detected at that time. So this was the main problem. And if it is not integrated into the host genome, then with the passage of time, it may be lost by the subsequent cell division of the host organism. Why it is lost? The reason was that such foreign DNA fragments, they were not having any origin of replication that helped them to divide or multiply independently of the host organism. So that's why they were diluted with the passage of time with subsequent cell division. And if it is integrated, then in most of the cases there was no problem, because now it will multiply with the host organism after integration. But even there were some problem that how to detect this integrated foreign gene or the DNA. So some techniques were required to differentiate between the foreign DNA and the host genome. And then with the passage of time, this problem was solved.